Hello, everybody. Welcome. Thank you so much for coming to this morning session of the Cancer and Evolution Q&A session for Ken Pienta. We appreciate you joining us today. I'm going to pass it over to Ken and let him take it away.
Well, good morning and afternoon to everybody, and I really am appreciative that you would join this session to talk about in a little bit more detail about some of the things that I did talk about during my session. And you know, the main thing that I really did cover during my session, even though my title was Convergent Evolution and the Origins of Lethal Cancer, and I have a lot of thoughts about those topics. I really spent most of my time trying to convey the message that I think this concept of a transitory cell state that we term polyaneuploid cancer cells is the reason that basically all lethal solid tumors go through this, are all therapeutically resistant to systemic therapy and all metastasize. So for me as a clinician, I'm really worried about and thinking about all the time that even though cancer starts as an independent entity in all of our patients, and we've been especially reminded of how generally we think about disease as something infectious and that can be spread from person to person. That's not the case for cancer. And so how is it that every cancer that's killing 10 million people a year is arising and leading to this series of events that leads to metastasis as well as therapeutic resistance? And that's the two convergent properties that kill somebody, and the fact that they kill somebody is the fact universal. So how is that possible? And I've spent my life trying to understand that. And this recent, for us, recent appreciation that these polyploid giant cancer cells exist as a state inside cancers and appear to be a uniform transitory state for survival is what I spent the majority of my time talking about. So the key, just to repeat it for us, is that for cancer cells to survive, the insult of, for example, chemotherapy, they appear to go go through whole genome doubling and go through an S phase that allows them to double their aneuploid content, which is why we call them polyaneuploid. And then they exit the cell cycle and go to basically be in a G0, G1 state, which in normal development is called an endocycle. So what's interesting to us is it doesn't appear that an aneuploid cancer cell can get to that quiescent state without doubling their genome. It's like they have to enter the cell cycle, and that's one of the things we find really fascinating. And then when stress is removed, I spent some time thinking about how these cells sort of pass resistance on to their progeny, and we talked about the different models of that. So I'll stop there and see if anybody has any questions or comments, and we can kick this off.
Yeah, so if you have a question, feel free to pop off of mute. We're going to keep it pretty casual today. If for any reason you cannot unmute, feel free to put your question in the chat and I will read it aloud.
Ken, this is Carlos.
Hi.
Hi. I don't know if I understood you correctly when you said that cancer cells enter continuously into the cycle. Is that correct? Is that what you said?
So, basically all cancer cells are aneuploid and are, proliferating and proliferating fairly rapidly. And what we have noted experimentally, and others have talked about, is that when a cell experiences stress and it's trying to protect its DNA, basically, what the cell does is it goes through an S phase, doubles its whole genome content, and then exits the cell cycle, the proliferative cycle, and enters a G0, G1 state where it's paused. It pauses its proliferation until that stress, that toxic stress, is passed. And then eventually it comes out and re-enters the cell cycle to undergo depolyploidization. And so what we're trying to sort of tell the world is that cell state, that transitory cell state, is actually an extremely important but underappreciated moment in cancer cell tumor survival.
But isn't it true that the process by which we can find a tumor takes years, and the proliferation of those cells is rather short. I mean, let's say slow, because most of them do not proliferate, and those who do it regularly, and as it's well known, it takes decades sometimes to find and feel a tumor, right?
So if you look at the, it is generally thought that to find the first primary tumor grows up over years and maybe decades. And if you look at the proliferative rate of solid tumors, even at diagnosis, and this has been, you look at the S phase fraction, it can be anywhere from 3 to 10 to 15% at the most. And in general, that is balanced by an apoptotic rate that is very close, so that the tumors grow, at least as a primary, very slowly, because there's a balance of proliferation and death. When you have a metastatic deposit, those numbers the proliferative rate tends to go up, and the growth rate overall for the entire tumor is faster. And whether that's a combination of higher proliferative rate as well as a lower apoptotic rate. Again, this, under normal circumstances, quote unquote, unless the tumor is stressed, the cancer cells are stressed by an unfavorable environment, or whether that's an external pressure or an internal one, like the tumor microenvironment, low oxygen, you don't need to form these polyaneuploid cells. They are specifically a stress response state.
Could I put in a point on this issue?
Please.
Of speed, because I like what Carlos is getting at very much. Sure, from one point of view, we see the possibility that a cancerous state might exist for 20 years before it explodes. Now, in your abstract, Ken, and in your presentation, you drew an analogy with the Cambrian explosion, which of course is one of the obvious analogies in looking at how ideas about evolution could influence our thinking about cancer. Now, I'm going to make the obvious point, which is that on the timescale of the Cambrian explosion, 20 years is nothing. So the analogy with the time taken for a group of cells to depart from the standard, if we can call it that, or normal form, to first of all being different and therefore triggering stress, immune response, and so on through being different, that time, it seems to me, it can be almost any length of time, can't it? I mean, the analogy with the evolutionary process here, I think, is illuminating. And now the big question is, how do we distinguish the ones that can radiate so rapidly from the ones that don't? And there's a big issue which I don't see anybody having during the conference have addressed with new data or new ideas that really tell us the answer to that question. There was indeed, there was a question in the, after my talk, which referred to Carlos and Anna's idea of the field theory of cancer, which is how on earth does it account for the fact that some cancers don't take off, others do. And do you have any insights yourself on that question? Because it affects enormously, doesn't it, the wish of people like Azra and others, in fact, all of us, to find ways in which we could mark the early stages. And it matters enormously then to know whether this is going to be something that explodes rapidly or very slowly.
Yeah. I wish I spent a lot of time thinking about that, and I think there's no simple question, but it is the fundamental question. Henry wants to say something here, so I'll stop and let Henry chime in because I think he go ahead, Henry.
Hi. So can you hear me?
Yeah.
Okay. So also to answer the Carlos the question. So the trigger the system chaos. This process depends on different model can clearly reflect the speed. For example, from the immortalization model, we actually in vitro culture. So this whole process, from trigger to gradually accumulate the population is take about 3 years. But when the genome becomes extremely unstable, like drug resistant, always achieve in 3 to 7 weeks. So what's the difference between the initial cell, right, that become immortalized and goes through this different circle, is the genome instability themselves. So we did an experiment to answer the Denis question. So if you with a p53 lockout, then you are very easily trigger the random chromosome underjoin, right? So it's very easy. So, but we block this pathway. For example, we block the non-homologous random join pathway. The incident is reduced, but still exists. So life always find another pathway to compensate what we are doing, right? So the idea is in the normal people, normal cell, if you by chance to accumulation the change, you want to reach to this chaotic point, you need lots of time. So for example, for the BRCA1, BRCA2 mutation, you have a much higher chance to induce the genome chaos. But for the normal cell, if you induce genome chaos, you try to kill them, most of them, they cannot go through this pathway. They just simply be eliminated, or the checking point just simply stop the cell growing. So I guess one of the key information we're still puzzling is, under such a high stress, so it seems like the information try to survive. And actually it's not a kind of simply random create such a motion, but just do whatever you can have a new information. But after that, Carlos, you're right. 99.9% of the cell cannot survive. We actually monitor the process is always dying off. Only few on the perfect storm, they form the cancer. But because of the system is very aggressive, so that's very easy to genome change. So the survival, the frequency become much higher. So we did the quantitative, semi-quantitative study. So for example, if you use, the small flask, you see this come back, this rate. You need to repeat almost 100 times. We did multiple times. The smallest number is 67, highest is 100 times. You see, you keep repeat this process, then one of the survivor will become dominant. So then we actually change use big flask, because number is a potential of really. Then you become 1 in 10. But for the patient, if they have a billion cell in the cell, I mean, if you trigger this whole process, the chances are they come back every time. So that's what we're thinking, the number game and also the stability themselves. So that is extremely important. Anyway, so does that make sense to some of you?
Well, I think it does make sense, and it gets to Denis's point that we do not have a way to reliably identify, certainly in vivo in a patient, when that event occurs that takes you to a place of lethality. We can't find that first cell that does that. Which is Azra's goal, and all of it would be great for all of us to have that goal, but we don't have a way to find it, and we don't have a good way, really, in a person to measure, in real time, what are the important steps in the evolution of that tumor, and when it becomes. When it picks up that lethal phenotype, whatever that lethal phenotype is. I think in general, we don't even, in a tissue culture system, we don't have great ways of monitoring actual death, right? I'd be interested, Henry, for you to tell me how you're actually, measuring that 99 clones die out of 100, because it's really hard to capture those events. You can, sort of measure, an X and 5 and say this many are on a path, but when you have a mutation that supposedly endows, or that changes the cell's ability to survive, we don't have good ways to measure the ones that are dying. And so we don't know the efficiency truly of genetic instability, which I think is a major problem for us to understanding cancer evolution in a person in general. Is that we can't measure what's not working. We talk about it all the time, but we really I don't see anybody measuring that.
So what we have done is, you just monitor the in situ single cell diluted single cell pass through generation by the moving, and every time you see the different clone, and then we analyze each clone the genome type. So initially, after 48 hours treat, almost every cell you analyze have different chromosome composition. But then after with a week to go in, the majority of them be eliminated. So all of this crazy stuff is transitional information. So then if you just use one flask, usually they have become one cell pop, become dominant. But if you use 100 flask, they actually usually have 100 become dominant. So we know is an opportunity to let this process to play out, to tell us the story, right? So that's actually the thing. But to a further question is that is to say whether or not some individual is more unstable than other. The question is yes. We actually measure the blood to say random chromosome change. Different patient is totally different. So we try to promote the idea in the future where we're screening patients before the chemotherapy. So someone with unstable genome, we should avoid the drastic stress the approach. So we also did some kids, trace the kids during drug treatment progress to see the genome stable or unstable. It's not a cancer's genome, it's the internal, the individual's genome. So there's two concepts. One is you direct opposite the tumor to see how heterogeneous they are, but another way is a constraint. So that could be interesting, like a case for, we actually try to understand that for those people quickly develop drug resistance, whether or not those people's genome baseline is very unstable. So that's another question I threw them out so we can discuss whether or not it's possible.
If I try to get to some kind of understanding, isn't it true that my small intestine cells don't cease to proliferate and they never get a tumor? Isn't it true that in psoriasis, there is incessant proliferation and there is never a tumor? So aren't we, let's say, barking the wrong tree, looking at cell proliferation as something that causes cancer, when in fact is a consequence? All the things that we are hearing about what's wrong with cell proliferation are consequences of cancer. They are not the cause of cancer, because cell proliferation is universal for normal and cancer cells. I am unaware of any evidence that indicate that all cancer cells results in triple cells instead of double cells. Out of a normal cell, you get 2. Out of a cancer cell, you get 2. So I don't know what the big deal is. At least.
Well, I appreciate that, and actually, it's something I've referred to as the proliferation paradox. And the real issue here is not proliferation, but where that proliferation is happening. I noticed Emma Hammerlund's on the call now, and she and I recently wrote a paper about this exact issue, and thinking about, in evolutionary terms, like Hydra is constantly proliferating, right? But if you think about Hydra, it's very similar to a small intestine, and what's happening though is that you're sloughing those, it's constantly proliferating, but you're also sloughing those cells. In psoriasis, you're proliferating, but you're sloughing. So those cells never have a chance to actually form a tumor because they're sloughed off. Your hair is constantly proliferating, but you don't get hair cancer because it's constantly getting out. Whereas in the colon, a much lower proliferation rate where you don't slough. You have to put it in context. You have to have proliferation, but proliferation, has to be put in the context of the tissue, as you have, pointed out so well. And it really becomes an issue of tissue, right? It becomes an issue of where is that proliferation happening and how is it happening and what else is going on around that tissue, that also. And then there's proliferation rate, right? You can get a lipoma that is as big as anything can be as big as 6 kilos, but it's growing so slowly and it never picks up another mutation that allows it to become cancerous. So this, again, it's way beyond proliferation, because you have to get to the point of what causes the lethality. Proliferation itself is not the lethal event. If you only proliferate, you can cut it out and get rid of it.
But Ken, you almost got it there. You said it's a tissue phenomenon. Now, that will make Carlos very excited, of course.
Yes.
That's his theory. Now, having excited Carlos, I now want to damp the whole thing down a bit, because, yes, that gets at the reason why proliferation itself is not the issue. And I thought you beautifully explained that. But that gives a clue, nevertheless, doesn't it? What in some tissues puts rogues under stress, and in other tissues that stress is absent. What is that?
Is that a question for me?
Well, go ahead. I have an answer too, but go ahead, Carlo.
No, but this is your show, so I don't want to.
It's a conversation.
Oh, okay. That is a very relevant question to ask. The issue is that is not what we are talking about. What we are talking about is what is cancer and how cancer is generated. The issue is that as I said, for example, the argument that cells in the small intestine slough off, but the cells that proliferate, they proliferate at the base of the crypt. So this is not a problem that happens at the villi level. The proliferation starts at the base of the crypt. So the proliferation is incessant, and you never get a tumor in the small intestine. That is a fact. You get proliferation in the skin of psoriasis people, and you never get a cancer. So I'm sorry that Perry is not here, but as a physicist, he must remember what Richard Feynman said. You guess, you test, and if the test comes negative, you drop the idea. Your guess has been wrong. The issue is that nobody wants to drop this test, that proliferation is central to cancer. Believe me, I have been growing cells for over 50 years in culture, and realized that most of the things that, for example, Henry has been describing are artifacts. I mean, to talk about the immortalization, that is a creation of, is an anthropocentric creation. I will die and my cells will keep on proliferating. So that is the basis of why we have transplants. We use the transplants of cells that from a guy who died. So this idea of senescence, cell senescence, is an artifact. And people, of course, make a living out of it. But I am not the one who is going to, let's say, rain in their parade. I mean, everyone has a right to have a job. The issue is that we are talking science, and we think that there are principles that you have to respect.
Sure. So actually, could I just respond? Because so quite interesting. We all thinking the tissue is very important because But on the other hand, we have to answer the question which.
That's a novelty.
Which is listen to me to explain to this one, because you are actually we all somehow had our own bright spots, everyone including. So we just try to see from different angle. So the cancer initially somatic cell mutation emphasizes the proliferation by the wrong reason, which is not the reason. The reason is if you are Darwinian evolution, so you have one new mutation. If you want to gain the second mutation, you have to have certain size of the population, then you have a chance. So that's the imagination. I disagree. Carlos, just listen. I disagree this way because that is the way people propose the stepwise, like 4-5 gene mutation responsible for such a thing. But on the other hand, people always ask the question, what is cancer, right? As I just point out, if you focus on overgrowth. You're thinking cancer is a problem of overgrowth, which we disagree. But if you study development, you always say, oh, cancer is a problem of cell differentiation and de-differentiation. So if you study mitochondria, you say, oh yeah, the cancer is a problem of the metabolic sources. If you use different sources, you have cancer. But in fact, what's fundamentally cancer is a new system emerge. Then you are asking me, what is a new system? Right? You tell me because cancer and the normal have same genome. This is not a fact. They all have genome change, right? So this is, so bear with me. You all have the genome change. So, but according to our so-called two phases of evolution point of view, so you initially genome reshifting, but then after that, when you become cancer, you need a perfect storm to this cancer cell gradually growing. That's a factor too. So in this case, the gene mutation, because if you sequence different patients, they all have different gene mutation. So the gene mutation could be stochastically hijacked by this whole process to help cell population to go in bigger. So that's basically the idea we try to put there. As for this tissue culture is absolutely artificial system. But what they tell us is the evolutionary pattern in the biological process. The good news is after sequence all the cancer genome, the people talking about the Big Bang, initially all the copy number drastically change, and gradually that's universal for all the cancer type in vivo data. So that's the idea. So we are not just talking about simply use for example, drug resistant. It's also artificial, like we're doing this the culture. We put the drug, we see how it's come back. It is artificial. But when the data is fit, the in vivo or the patient's data or the animal's data, you have to think about whether or not they have some channel for us to gain the information.
May I answer that?
Sure.
Okay. Henry, I assume that you read the literature like anyone else. And in the last year and in the last month, people have shown clearly that there are a lot of clones of mutated cells in driver cancer cells that never get into a cancer. What else do you need to drop that idea that mutations cause cancer? I mean, this is not data that I generated. This is data that people generated at Johns Hopkins and at the Dana-Farber. So it's not my data, it's their data. In fact, what happens is that they did the experiment in reverse. Instead of using a control of normal cells to see whether they are cancer mutations, so-called cancer mutations, they started with the mutated with the cancer cells, and then they came to the normal. And the normal showed that the same mutations that happen in cancer cells exist in normal cells. That is a perfect idea to say, let's drop it. We had a century of this stuff. What else do you wish? $250 billion. This is Astra's numbers. What else do you need?
So I don't disagree with that, but you have to also, accept the data that cancer, all lethal cancers are aneuploid and have multiple mutations. And you have to get to that point at some point. You have to get there. Proliferation is a piece of that. I don't believe that it happens in a stepwise fashion, but what you have to, let's talk about what kills a patient with cancer.
We are not talking about that. We are talking about how come that mutations are in cancer cells. And I think that is an epiphenomenon. And if someone has a different idea, they keep on, I'm not saying that a lot of people think like we think. A lot of people think like you think. And of course, this is how you keep on doing the same stuff. looking for mutations that are present in normal cells that happen to be present in cancer cells.
I have to agree with Ken in such a way is when we say the important clinical fact, we have to think about, we cannot ignore them, right? So as such, so for example, Carlos, my writing for past over 20 years. I strongly against the gene mutation theory of cancer.
It doesn't show.
Exactly. But then, at another hand, the fact is the patient also have lots of gene mutations. So we try to use this. This is also, we need to explain. We cannot just completely ignore that. So therefore, so for example, the Ken's exposure, the new information, and the later stage, whether or not the gene actually plays a role for the population growth, that's a very legitimate question. Of course, we did design this experiment to showing that, but from Stanford group's data, when this early stage of the primary tumor evolve, so the cell, metastatic cell already hiding there, but those dominant cells that do not have the chance, that could be if they did not have hijacked some of the gene mutation to help them clinically. So that's just the hypothesis to thinking, but you can just delay this whole fact or observation, but it's interesting to study. But just for you, I really want you to reread Mead's paper. She clearly said without a chromosomal change, then the cell can transform the phenotype. But with it, it's no way to do it. So you need to reread this 1976 PNAS paper. And later on, 1980s, she did the karyotype analysis, found all the tumor cell, which she used, 95% of them is normal chromosome. And then in 2004, Harvard group repeated experiment found that when you have chromosomal change, you cannot reverse the phenotype. This 3 paper, you read it, then we can discuss, right?
James Shapiro has a comment.
James is going to say something.
Well, Ken, you go ahead because it's your discussion, and I'd like to just make a comment.
Please comment. Go ahead.
Well, I just wanted to say to Carlos and to everybody that we shouldn't think about mutations and other genome changes as equivalent. And we should also not think of them as stochastic events, but events that are subject to influence by the conditions under which the cells are growing. And if they're stressed, they make more drastic changes than if they're not stressed. And Henry's point is that a chromosome reorganization is different from a mutation in a driver or a tumor suppressor gene. And we know that all of these things happen. We can document them in tumors, and they play a role, but they're not necessarily the whole story. And I think it's the responsiveness of the tumor to its tissue conditions, but also to be able to restructure its genome internally that play a role in the formation of cancer cells. And we have to figure out exactly what those connections are and what the triggers are so that we can intervene in blocking them somehow.
I certainly, Jeff, did you want to say something? Okay.
No, sure. I mean, if you want. No, I would totally agree with what James said. Like the thing is that I do feel like there's a lot of sort of like, let's draw a line and say this isn't true or this isn't true. Like all of these points need to be taken into account, and we need to think carefully about how they all integrate. Like it's, obviously the mutations play a role, whether it's hijacking or whether it's just sort of epiphenomenon, they play a role in the phenotype of that cancer. So we don't need to sort of discard them. What we need to do is try to come up with a more synthetic way to account for all of these different things, just like James was saying, so that we can understand what the common effects are and what the temporal stages that go along in this cancer evolution are. And hopefully if we can figure out those temporal stages of whatever the properties are that are really critical to cancer development, then once you have an idea of staging, and now I know like there's some concern about, and actually one thing I would like to have a better understanding from you guys is there's a lot of, it's not stepwise. And I sort of understand the rejection of a stepwise model as a very simple caricature of a model. But on the other hand, as scientists, what we're trying to do is understand what the steps are. At least that's what I understand. Am I wrong about that? I mean, I want to understand, A happens, then B happens, then C happens. And let's not name what A, B, and C are, because everyone has different ideas of which are the critical roles of those. But ultimately, what we want to know is, what are those steps? And so ultimately, I think we should be very careful about saying, look, this is a thing that we want to understand how it's happening temporally in this process. And let's talk about how those things interact. And then one other caution I want to say, just as someone who's sort of coming from a slightly different perspective from everyone else here, I think, is that I do think that essentially, like, the framework that one thinks about this in terms of evolutionary theory is appropriate for any such bits of steps that we put together. So it's true, we may have to think very much out of the box in terms of what those steps are. But the basic idea is you've got something that is passing on some heritable trait, and whether it's a gene mutation or whether it's an epigenetic phenomenon or whether it's whatever it is, it's passing on something that retains its identity. And that retention of identity is something that we understand as an evolutionary phenomenon using kind of standard evolutionary theory. We just have to be much more open-minded about what those states are as we think through what's happening in cancer.
Yeah, I appreciate that, Jeff and James, and as my mentor used to tell me all the time, everybody's right.
No, that is not what happens in science. Someone is
true. Not everyone is right, but
otherwise, I spend my life in an endeavor in which everyone is right. So what is it's like dancing with your sister.
Okay, I won't go there.
I think we could safely say everyone has something to contribute, and
okay,
maybe
that's something else.
But what I was trying, one of the things I want to come back to, and I was trying to draw attention to in my talk, is the fact that we've all grown up, so to speak, or been part of the hallmarks of cancer. And whether you believe those happening stepwise or believe in them all, what I was also pointing out was that you can't explain universal therapeutic resistance by what appears to be a simple mutation model. The fact of the matter is that cancer cells, a tumor, whatever that population of cancer cells is there is some population within that tumor that's resistant to stresses that they've never seen. So, that the reductionist approach has been, well, you upregulate this drug pump in one cell stochastically, you have this repair mechanism in another, but just I've never understood, or I've come to not understand or be able to explain universal therapeutic resistance. And so we have become to believe over the last certainly 18 months that this transient cell state that we call the polyaneuploid cell may be a mediator of that therapeutic resistance, because we see it in our artificial system of, if you kill, in cell culture, all, put in a LD99, that's all that's left are these packs. And they repopulate to a 2N plus aneuploid population. We have to explain that phenomenon, and it doesn't matter what stress we use, they come out every time. And I don't think the cancer community has appreciated that. I spent 30 years of my life not appreciating it. And yet I think it's so fundamentally important. I've changed everything we do in the lab to try and understand that cell state, and how it then, I don't know how they end up mediating, passing on resistance to their progeny, but it looks like that's the universal mechanism. And, I need that explained to me. It's changed the way I think about cancer lethality.
Well, if I could intervene here, people have shown experimentally that if you mess up cell division, you get polyploid cells.
Right.
And then if you let them go through their aberrant cell division and produce pseudodiploid cells, you get cells with scrambled genomes and new properties. So that we know happens, and it can be induced by cancer therapy agents. So it may be that the resistant cells do not necessarily pre-exist the therapy, but the therapy can actually trigger their formation.
Right, but so what we're trying then, I buy that. 2 points. The first point is the cancer community in general, meaning that 99 out of 100 people studying cancer don't recognize the polyploid phenomenon. That's number one. Number two, certainly one of the things that has been bothering me, and again, I can't explain it, and I tried to touch on it in my talk, was the fact that when we see the resistant population progeny sort of grow out and they have that resistance, we don't see concomitant death of a bunch of other clones. So we don't see this random sort of genetic instability, finding the right clone and that one growing and 99 dying. What we see is this paused proliferation, and then all of a sudden all the clones grow out and none of them are dying. And that's something that I want explained and what we're trying to explain.
Are you tracking the DNA on those clones?
Well, now we know to do so, and that experiment will happen over the next 6 months. We didn't know to do it 6 months ago. We know it now, and we're doing it through our plan is to do it through a combination of lineage tracing with single cell analysis.
So to put a little bit more, so we actually, the King asked a question when I give a talk, I never got a chance to answer, because we thought that this whole process, the chaotic process, is deterministic. So the King said, how you can say is deterministic in such a way? Because this whole process, they have 3 major steps. First step is stress. Regardless of what kind of stress, because over 10 years ago, we did the same phenomena. We say, what kinds of molecular stress can trigger specificity of the response? So we use a chemotherapy, we use molecular drug specific therapy, we use switch temperature, we use change the nutrition components. Same thing, they can trigger the phenomena. So we know the trigger fact is stochastically or randomly in such a way. And secondly, when they involve the genome chaos process, the process themselves also have lots of randomness in there. For example, they can go through the polyploidy. They can use the massive translocation. They can form many of the micronuclear clusters. So that's why, this also looks like it's very stochastic. And then finally, for the winning cell, if you repeat many times, we repeat many times, those same time have same karyotype. They all changed different every time, right? So say the major 3 steps, they all have lots of stochasticity. But one thing is for sure is under stress, the cell favorably is reshuffling the genome, and with the number game, they all succeeded, either become drug resistant or become killing power. So that's the process is very much certain. With certainty, if we have enough cell, you restrict, they always get to this one.
Yeah, so I appreciate that, Henry, and I can buy that genome chaos leading to aneuploidy and chromosome rearrangements. The whole how you generate that the aneuploidy, I buy. What I can't, or what I don't see, again, it's in cell culture, it's an experimental system, but when I generate polyaneuploid cells, and I don't see genome chaos. What we see is whole genome doubling with no rearrangements when we do single cell. So basically we see the 2N plus the aneuploid population shift up to 4N, 8N, but we don't see chromosome rearrangements in that setting. So there's this cell state that we call polyaneuploidy or PGCCs, whatever you want to call it, isn't doing the genome chaos thing. I think genome chaos can happen and does happen. I mean, I think you have to you're generating this aneuploidy, but I don't see it in this cell state. I'm not seeing it as the resistance mechanism.
Sure.
And again, that's in a tissue culture experimental system, but it seems to us, again, what we see is the doubling of the genome, the paused proliferation, and then the resistance. And we have to do, the experiments that Jim's talking about, which is to track them individually. But at the level of the genome, we're not seeing those rearrangements in the packs. And that's just the data. And
Sure.
so I'm left trying to explain the data.
Sure. So if you understand, so when we talk about genome chaos, the translocation you say is structural chaos. Numerical chaos is always genome becomes so many. So if you're thinking about the so-called the karyotype coding. So either structurally change or number change. They just change the genome gene interaction network. So that become totally new system. So in the mouse, we found that it's very often, like just chromosome numbers shoot sky high. But in a certain stage, they will trigger out this mechanism. For example, in the flower plants, initially by the doubling the genome, like everyone thinks the genome. But at a later stage, when under the stress, the differentiation into different species, then the chromosome reshuffling becomes a major force. So if you're looking for the evolution, not just our cancer system, but for this, so it's kind of universally goes through this way. Regardless of what, the change the number of the chromosome is not just a gene dosage event, it's a coding change, and which trigger further. Yeah.
But wouldn't you see that with CGH?
You cannot say it unless the single cell CGH, because CGH is a way to say population. They wiped out the average stuff. In fact, most of the random change is below the 5%, and that have been wiped out by CGH methodology by the normal sequence approach. So, but the cancer is a game of outlier. Just a few outlier that trigger the whole system.
Yeah, but in this setting, it's not an outlier event. You're not seeing an outlier event. You're seeing doubling of the genome, pause proliferation, and then a resistant progeny. That I don't understand how you would say that's an outlier event. Because there's no bell-shaped curve there.
So in the cell culture situation, all the cells have enough nutrition. They don't worry about all of this. They don't have competition. So in the real world, you have so many changes, but only few can survive. So that's the story in the vivo cancer situation. Most of the change don't have a chance to be successful.
If I may come back to
James and then Carlos.
Yeah. I'm just saying maybe in the system you're studying, you're getting epigenetic changes. And you don't see those in the genome, but you'll learn that when you look at the DNA and presumably look at
we'll do single cell and we'll do RNA and CGH and the whole works. So we'll have an I think we'll have an answer to a lot of this 6 months from now. But Carlo?
So the Jinsong Liu, they did lots of sequence that are going to publish soon. I mean, every time they say it's completely changed, but could be the system, in different stage. So that is fine because, as I said, all this, the way to change the coding should be achieve the same purpose, which is be different. So the cancer just be different, nothing else. Yeah.
Carlos, go ahead and make your comment.
Yeah, I wanted to come back to a comment that Jeffrey made about the cancer mutations, the role of cancer mutations. And I wanted to ask him or whoever, what is the role of cancer mutations?
Well, look, I mean, that's a huge question.
No, it's a very small question. You tell me which is the gene that causes cancer.
Yeah, I mean, I'm a little worried about derailing Ken's Q&A if we have a long discussion.
Sorry, you have the right to.
No,
but I'm happy to.
We're good.
Okay.
Go ahead.
Okay. If you're good, Ken, I'm happy to.
I'm good.
Question. I mean, you know, they all have very different roles. I think, like, let me try to take the 10,000-foot view of how I think we need to look at mutations and understand their role in cancer tumors. When we go to, you know, and what I do in my research, we're looking at this genomic data and the same data everyone else is publishing papers saying, you know, p-value 10 to the 10th, this is a cancer gene or whatever. You know, what we're doing is looking at many tumors. You're going to, you know, take a piece of tissue from a person and another piece of tissue and another piece of tissue. And we're asking the question, what is the frequency with which you see particular mutations in those tissues? Now, hold on a second, because I know what you're going to say. You're going to say, well, you see these in normal tissues too, right? Right? I don't know if you said it. Actually, it's really important to look at what they are in normal tissues because there's some combinations of mutations that appear to happen in normal tissues, and there's some combinations that happen in cancer tissues. And we haven't sorted out yet. We have not answered the question that you point out, right, or have pointed out earlier today, which is like, how do the combinations in normal tissue differ from the combinations in cancer tissues? That's not an answer that we have. And maybe they don't, in which case maybe the mutations are all epiphenomena, as you say, but it's not also proven that they are the same combinations that you see in these different tissues. So we need to sort that out. And I think it's very much an open question. I think there's reasonable reasons to believe that mutations do play a role in cancer. And in particular, the thing that I would say is that there's not a reason why we do have pretty good estimations of the underlying mutation rates of particular mutations that appear in cancer. So we can get an idea of how frequently this particular base pair happens just in the tissue that is the basis of melanoma or the tissue that is the basis of colon carcinoma. So when we know that baseline mutation rate, when we go and sequence a bunch of tumors, there's no reason to see an overrepresentation of that mutation in the cancer unless it has something to do with the actual cancer development. Now, how much it has to do with the cancer development and how much it accelerates or causes cancer is still an issue because that depends on one of the things that Henry Yang just mentioned a bunch of times. There's so much stochasticity in whether that mutation actually helps the cancer to develop that way or doesn't, depending on whether the cancer is under stress, depending on whether it has enough whether the genome is messed up enough that it can actually work. Like there's a lot of other dependencies that play into that. There's a lot of stochasticities, which means we don't know the population size for which it is true that a mutation in KRAS G12 actually, has an impact on the development of cancer. We don't know that, unfortunately, because there's so many stochasticities. And the point is the stochasticity that actually drives down the number of these things that actually make a difference. And but in the few cases where you have the right environment, the right, et cetera, presumably it's making a difference. And the reason I would say I do believe it's making a difference is that the mutation at the end of the day, we're seeing it at a higher frequency than we should on the basis of the underlying mutation rate. And there's just not another way to explain that except for that it's actually, is actually in the developmental process of that cancer as an entity that has to happen. Now, whether it has to happen because it's a consequence of something else that has to happen, or whether it's causing the cancer, we could delve into more debate about, right? So, I mean, if you say this is an epiphenomenon, then it could be that, A and B and C happen. And as a consequence of B, you have to have that mutation. But it's sort of, you end up into some very interesting causal chains when you say, because of this thing that's happening, because of stress, et cetera, this mutation has to happen. And yet it's not important for cancer. It gets hard to explain it. Now, and I can't go through every possible example, but that's the sort of set of reasoning that I would apply as a general rule of why we think that these mutations do play a role in the development of cancer.
We have a question in the chat, which is, Kenneth, you described large PACC cells 8 times the volume of typical cells, which can survive chemo by going into quiescence, which I believe meant a period of stasis rather than a normal cell cycle multiplication. But you also said that PACC cells multiply under stress. You stated that 30,000 could become 300,000 after treatment. At what point do they multiply just after quiescence?
Paul, thanks for that question. And that's a little bit of a confusion. That's because that's not what I meant. So what happens is that a sort of a 2N plus aneuploid cancer cell, when it is exposed to stress, some percentage of the population. So if I have 1 million cells growing in a tissue culture dish, at any one time, about 30,000 of them are PACCs, PACs. When we go ahead and put in a lethal dose, a 99% or 90% lethal dose of chemo, we don't see just 30,000. We'll see 100,000 or 200,000 cells, but it's a combination of the packs that were already there, as well as these 2N plus cells that go through an aborted cytokinesis. And then, so they go through whole genome doubling and then enter G0, G1. So that's how you get that increased population. There's no proliferation of the packs per se. They're not proliferating. What happens then after the stress is released is that you get a depolyploidization event where those cells start to go back into the cell cycle and divide. But so it's after that happens, after the quiescence, either when the stress is gone or they've quote unquote learned a way to get around the stress. So the actual formation is not a proliferation event, it's an aborted cytokinesis event. Does that make sense?
That's great. Thank you very much.
Oh, okay, good. So you know, I think that the real question we're dealing with, and you know, again, mutation versus not mutation, whether they're, you know, I don't think any of us on this, you know, believe that it's just a linear stochastic accumulation. But we do have to, you know, again, explain that cancer lethality is more than proliferation. It's more than these events, because you have these two phenomenon of, you know, metastasis and therapeutic resistance. If a cell never metastasized, you could cut out all cancers and they'd be cured. And I think all cancer biologists understand that. You know, when we talk about it, you know, for the evolution folks to just make sure they're getting that. But we also can't really explain why metastasis happens. You know, we've tried to use optimal foraging theory. We've tried to, you know, apply ecological ideas to that. You know, this idea of why, you know, how a cell leaves the primary, quote unquote, why it leaves the primary without anthropomorphizing any of it. And I see Jeff wants to say something here, is a key point. You know, one of the things that we've been trying to deal with is you have metastasis, you need that to be lethal. You have therapeutic resistance, you have to have that to be lethal. The question is, are the two related? Is another piece of that. Jeff, please go.
Yeah, I just wanted to comment that at least one, you know, at least partial possible resolution that goes on between the sort of somewhat differing opinions like on this call is that what has been true is that despite tons and tons of all the sequencing data, we are not associating mutations with metastasis, right? Like, right. No matter what we do, we don't find mutations that lead to, there's a few that seem to like maybe help it a little bit, but they're just like, you know, a little boost or something. They're nothing like the primary tumor things, drivers where you see KRAS G12 in almost every pancreatic cancer. I mean, like, it's just unbelievable. And it's not and that's not a mutation that should happen in your pancreas. I mean, there's not an environmental reason why you expect that to happen at that frequency. So there's, so one of the things that's really key here is that the metastatic transition seems to be something that, you know, it may be aided by mutations, just because the same mutations that help a primary tumor appear help metastasis to occur. But there aren't these, there was this idea for many, many years that we were going to find the mutation that confers metastasis and then we could stop metastasis with a drug. That is just not happening in that way. I mean, maybe we can stop metastasis with a drug, but it's not because of a targeted mutation.
Right.
We've done enough studies to show that. I mean, I published a paper in 2016 where we tried 40 different patients, tons of sequencing across multiple tissues to try to figure out in the metastases, are there any mutations that are leading to these? And there's nothing. There's just nothing in common among metastatic lineages compared to primary lineages. So, that's an area where I think we can all agree there's not really a genetic, evolutionary basis for metastasis itself. That's a much more developmental phenomenon that has to do with, the EMT transition, all of the developmental biology that I'm actually less familiar with. So I don't want to go into it further than this, but I do think that there's ample evidence that is a really, really key developmental point that is not really, it's not evolving by these kinds of mutations that most people spend their time looking at in cancer biology and cancer genomics.
Yeah, and it's also, whatever that the epiphenomenon of metastasis is, we also know that it's a very stochastically rare event. We've spent a lot of time looking at circulating tumor cells over time, done the calculations, and it's likely that it's to get a cell that leaves the primary tumor to form a clinically important metastasis is a 1 in 10 billion event. And what is that? What is that? And is it just that cancer cells drop into the circulation and end up with, occasionally making it? That's why we again think these packs may be important mediators, actuators of that, because they're both very motile as well as quiescent. And having spent 20 years studying cancer cell dormancy, and understanding the rat mind that is, this is another way to think about that. But Carlos, you had a question?
Yeah, I mean, a comment. I find it intriguing that people who are so concerned with cancer and evolution do not keep in mind the fact that proliferation and motility are the default state of cells. In other words, you don't have to stimulate cells. You have to let them do what they are supposed to do, which is proliferate, and what they are supposed to do, which is to move. So, of course, you will never find a mutation, because proliferation and mutations are constitutive properties of all cells. This is what we do when we put cells in culture. I take my fibroblasts, I put them in culture, and they, as soon as they are in the plastic, they start proliferating and moving. While in my body, they do not. And it is clear, this is 1912. This is Alexis Carrel, who said that what happens is that you disinhibit cells. And people who are concerned with evolution do not keep that in mind. And they invented growth factors and invented oncogenes, which in a way is let's say, an under way of doing, let's say, intelligent design. Because the growth factors that eventually, according to this old story, must have stimulated the cells, must have been put there by somebody, right?
I'm not going there.
No
way am I touching that one.
Darwin said that. This is not me. I didn't.
Well, Artie has a question, so we can ask that, because I think it somewhat relates to your comment, Carlos. In reference to the question of the role of mutations in tumorigenesis, has anyone considered that what we describe as oncogenic mutation is the actual stress? For example, when you try to overexpress an oncogene in a normal or even in cancer cells, the majority of cells die due to the oncogene-induced apoptosis, and only a few clones survive.
You know, Artie, that's a great point. And we've been, it's what, you know, oncogene-induced apoptosis is a phenomenon that I think cancer biologists accept and understand happens. You know, too much of anything, too much of a good thing is not good. And if you think about Joel Brown, has thought a lot about this, and you know, when he thinks about G functions in adaptive landscapes, and in that setting, you know, there's an optimum for any trait, and if you have too much of that trait, your population tends to decline. So you know, if you think about that, it's true. You know, too much of a dose of an oncogene can be a bad thing. You have to find an evolutionary optimum, whether it's for a cell or for a species. So absolutely right. I don't know if anybody else wants to comment about that, but that's certainly something we believe in our modeling with Joel now.
I could comment a bit, because people actually repeat the Bob Weinberger's experiment for cell transformation, the Japanese group, quite good group. They found unless the oncogene-induced genome instability, they will not cause this cancer transformation process. So back to the case, another question, because I want to ask you is, you're talking about you are the one of the person many years ago start talking about the cancer is the ecology, problem. So one of the key for the ecology problem, is different use adaptive landscape to describe this, is because you're dealing with different species. So that's the same. So the traditional Darwinian thinking different species is gradually by the population change the gene frequency. But now, since we know perhaps it's just when the chromosome changes, it already become new species. So that actually partially answer your question, why the metastasis, what's the difference? The Jeff said that there's no gene, but we know the new literature showing every time when they become metastasis, they have different karyotype, right? So then we need to actually validate if this is true or not, because even the George Vanderwood, all this publications, they actually identify some gene, but the title they say that is chromosome instability driving the metastatic. So the more and more people thinking about this way. But if you're thinking the invasive species, that's obviously is a different species, is not the gene cause this system, right? So how do you think in this one? If they are the different species, as you just proposed, therefore that code conflict with traditional Darwinian evolutionary study, the model, to solve this problem.
Just a small correction, Henry, to defend Charles Darwin. He also was a saltationist. I have the references for that. And I also often say that for a variety of reasons, Charles Darwin was never a neo-Darwinist. What you're referring to is neo-Darwinism, not Darwinism. Comment finished.
Okay. Yeah. So I see your point, but in a way that Darwin, even though from time to time he mentions the sudden jump, but for his theory overall is stepwise. So I always thinking to look to study people's theory, we actually need to see what's the overall framework rather than some individual incidents.
I think that overall framework was forced onto him by August Weismann, and then by Julian Huxley, by Fisher, by Wright, and so on, and we ended up with the modern synthesis. I think that's the biggest mistake biology has made in the last 100 years. But anyway, I've said all I want to say on that. I'll leave.
Yeah, I agree, and I'm waiting to modify. Yeah.
So Dennis and everyone, I actually think this is a huge point for the cancer field, because what I've noticed, and again, I'm just a simple physician here trying to play with ideas, but what I've run into full collision is this idea that cancer has to fit into the modern synthesis there's a whole bunch of people out there, the sort of the classic evolution folks that are trying to work on cancer that say you have to fit it into the modern synthesis. You can't talk about stress-induced mutation. You can't talk about self-genetic modification. You can't talk about anything that doesn't fit classic neo-Darwinism. And we're struggling with that. The field, the cancer community is struggling with this big time, which is, why I sent you that email earlier this week asking about the weasel. Because this is where, I certainly think things are going, but I'm running into the class, a lot of evolution folks that are working in cancer now who are saying, no, you got to fit it. So I'd love to hear your thoughts about that.
Well, I think this is exactly what Jim and I are trying to see to, of course, in the Third Wave Evolution group and in interaction, and in open and constructive debate with the neo-Darwinists. The problem is that the issues have been shut down for so long. I can give a list, I know James can too, of a huge number of people who were not listened to over the years. Barbara McClintock was not listened to. Conrad Waddington was not listened to. Carl Woese was not listened to. This is our problem. So I think we have to try to make sure that we break out of that framework. It's too restrictive, and it was not Charles Darwin's theory.
Can I add a comment to that?
Please do.
Which is that, Ken, you're going to write up a clinical review of the meeting, correct?
Correct.
And what I think I infer from what you said is that it's hard to get grants if you don't give a modern synthesis view of cancer evolution. And
it's impossible. It's pretty much impossible anyways, but again, you will not get that through a study section. No way.
Right.
If you don't ascribe to the modern synthesis, you're not getting,
well, maybe
get nerfed.
Right. So maybe it's time to use the meeting as a platform to sound the alarm that this is holding up progress in the field, because it's using outmoded ideas which don't incorporate lots that we've learned about evolutionary processes since the modern synthesis was formulated.
Yeah. I think that needs to be stated. I don't have the knowledge base to write that. I smell it, and I know it needs to be done. But in fact, we were, talking about, my group was talking about this exactly, that this needs to be done. But it needs folks like you to write it, or some combination, who really understand and can make the arguments, and who have standing in the field. For example, if I did a review and wrote that, everybody would go, he's just an oncologist, what's he talking about? And they'd be right. So, but this is, I can't tell you how important I think this is for, to move the cancer community forward. This is a critical point that we have to get out into. If I could be John the Baptist, I would, go out there and shout that look at this. This is the way it's going to happen. It has to happen. We have to think about this in this more global way, because it's holding. I honestly believe it's holding us up as a cancer community. I don't know what Jeff thinks, but I certainly.
Well, you don't have to be an expert on evolution to say that there seems to be a straitjacket on cancer research. You know about the problem with.
That's true. I know about straitjackets.
Right, exactly, and so you can say that, and there's lots of evidence. I just finished reading a paper about tumor cells and line elements, retrotransposons, restructuring the genome of many tumor cells, sure. Basically of 4 types of tumors, and there's lots of evidence in cancer, and there's lots of evidence in evolutionary biology. that other processes are going on and they're very important. And we don't have yet any comprehensive view of either of those subjects, but we do know that lots is happening which does not fit into the straitjacket which is being imposed on it by the study sections. And that is an identifiable harm which is being caused by narrow-mindedness.
Amen, amen.
So I would like to suggest not just amen, but another religious example. If Ken is John the Baptist, I think we appoint Jim as the Messiah, and I do the writing, and we get a transcript of this conversation, and we make it a centerpiece of the editorial aspect of the publication. We need to get action.
Yes.
I'd like to provide some of the historic information. So I'm so glad this time, because this time the Jim, Ginsen Liu, and the King, all this, me included, and Dennis, all we talking about we need to move out of the shadow of the neo-Darwinian. But I have been in the evolutionary meeting many times. My presentation basically the first half I have to present in 206. Every time we meet each other, we talk this. People just I'm the only person at that time talking about the non-neo-Darwinian concept. But this time I feel so exciting is because 5, 6, 7 people truly got the message. But unfortunately, for most of the neo-Darwinian research, they still didn't get it. They did not get it. So that's our duty to actually let them get it. Otherwise, they're going to continue doing what they're doing for another 20 years, and then that's it.
Well, this is a conversation.
Can I say something as probably the least enthusiastic of people to throw out, like what's happened in the last century in terms of evolutionary understanding? Just that my caution would be that if you use a lot of these terms, like if you talk about neo-Darwinism, the modern synthesis, talk about Darwin in general, an 1859 naturalist who is like long, that was a long time ago. What he thought is really not the issue of today. It really makes for a much more polarized piece of writing than it does if you try to address the scientific issues head on in the terminology of modern science. So I'm just giving you a little piece of advice here. Like, I think if you focus on those processes that James was talking about, and focus on what is being missed in the terms of the biology and in terms of the points that are important, rather than trying to scope it out as an attack on a sort of large and abstract philosophical statement about or a historical thing. I understand there's an attraction to that because you want to say, look, this is big and important. But I also think you're really, that is actually one of the things that leads to the sort of like exclusionary, or like sort of people just sort of saying, you know what, I'm not even going to listen to this anymore, because people are very invested in those things. And I think that where you can get much more mileage, if you want to, in the long term, really make a difference, is to explore those processes down to the last detail until you can say, look at this process. If you think this is like a process that doesn't fit within, the modern synthesis or within, what everyone is calling neo-Darwinism, once you have it, once you've explored that process or that stage-specific evolutionary motif or whatever your idea is, if you can develop it in a way that is going to be really, really strong and have predictive value, then at the end of that day, you're going to be able to say, look, this doesn't fit within within neo-Darwinism or within the modern synthesis, and everyone's going to just agree with you from a basic standpoint. So I just want to, the 2 points I want to make are, one, be careful about using all of these sort of like history of science terms as a way to motivate what you're doing, because I think it actually turns off the audience that I see you as wanting to affect. And then secondly, just because I've been participating in this conversation, I'm very interested in it. And I think there are a lot of really important ideas that need to be brought out. But I just want to caution you that, like, from my own perspective, I would be very happy to partake in some sort of discussion or a paper like this, but I'm a little concerned from my own perspective about whether I would agree with the statements as they are. So I would say, I'm just being very frank here, like it probably would be to the benefit of such a paper to have someone like me on it, who is part of the evolutionary biology community, who people view as a very stable, centered person in that community. And so I'm not, you guys can do what you want, but I just want to point out that it may be attractive to you to have someone like me on it, and to be on it, that would be, I'm just pointing out that it's like that's going to mean you're going to have to pass certain barriers of my own profession for me to be an author on such a paper. So I'm not saying anything about I have to be on or not or something like that, but I just want you to be aware of the strengths and the pluses and minuses of going different directions with what the conversation that we've had today are.
I think what you say is absolutely true. These are hot button issues for people, and they're sensitive to it. But there does come a point when you have to call a spade a spade. And there is nothing really radical in saying that molecular genomics can tell us about evolution and processes which were inconceivable in 1940 when the modern synthesis was formulated, and that they occur in evolutionary biology and they're real, and they occur in cancer and they're real. The whole Cancer Genome Project collaboration is based on that premise. If we can understand how cancer cells change, their genomes will have an important part of the cancer picture. Not all of it necessarily, but an important part of it.
I totally agree. I just think that the predictive value of the information that you're able to get out of an understanding of the process is so much better an argument for converting other people to your view than is the statement that this is not within some 1940s or 1859 person's ideas or 1920s or 1904 person's ideas. Like those are old ideas. And I think those people were brilliant people, but they didn't have as many shoulders of giants to stand on as we have today. And so when you set yourself in opposition to these old ideas, it has a certain sense of, oh look, I'm doing something important because it's different from what for 60 years people have been saying is true. And I understand the attraction of that, but it's a real danger because on the other hand, like a lot of people, I know, a lot of people look at it and say, like, why are you comparing yourself to someone in 1960 or someone in 1940 and what they thought? Like, I understand the historical attraction to it, but it has a lot of danger in terms of just setting you up as someone who's trying to aggrandize your idea by contrasting against something that's really old, as opposed to setting up your idea is really important because look, it has predictive value. It allows us to understand metastasis better. It allows us to understand who's going to come down with cancer. It gives us prognostic or whatever indicators it does. Like if we can, let's focus on the things that we really want to accomplish and their benefit, rather than, in my view, rather than contrasting against sort of old ideas that are these fixed mindsets. And the problem with so all those terms, and I'm sorry, I'm ranting now and you can just shut me down if you don't want to hear this, but the problem with like setting yourself up against the modern synthesis setter is that nobody has a very, there are different interpretations of what the modern synthesis is. And so the problem is that you can get into these really, really linguistic debates that I don't think are very useful, frankly. So, so I know there are conceptions that people have, historians have put together things, and there's very clear ideas. I don't want to, dampen that argument. I just want to say that it ends up going in that direction very frequently with those debates, where you're saying this is what they're saying, and someone else is saying no, the modern synthesis is something else, and it just, I'm not sure if it's useful to people who are suffering from cancer, just to sort of hook on to Azra's point.
This is very, very helpful indeed. If you read what Jim and I have written as books recently, you'll find that's precisely what we're doing. We're asking the question, what would you expect if you change the theory in the following way? What insight does that give you? I think you've written yourself, Jeff, into a role on this.
Yes, I was going to say that it sounds like you're volunteering to help draft a statement, exactly. which will have an impact and solve what Ken has pointed out is a very real problem in terms of getting research funded that might be important to cancer treatment.
So I'll just say, like, I would like to play that role. I do have, as we all do, have a lot of time constraints. And so I'm worried about playing that role in a very microscopic way as the thing develops, which might be the most useful way to do it, right? So to make sure that it ends up in the right direction. I think what probably would happen, just from a predictive standpoint of like what I think, is that we would probably put something together, and what the role that I could play most easily would be to look it over, say, look, these are the things that I think are going to fly or not fly from my perspective, and try to help. And then we would have like a decision point, I think, where we have to say, look, you know, like, are my constraints on what I think would fly or not fly like acceptable to you guys or not? And we would probably sort of have to sort of figure that out, and then I could play a key role in sort of trying to form it after that point. But it would be pretty hard for me to devote the amount of time I think I'd have to like work with each of you to try to sort of see how to make that happen. And so I just want to apologize in advance, because I think at that point, you can imagine I may end up sort of like, you know, some of you may be not happy with what I like or don't like. And I'm not going to, you know, from my perspective, that allows me the chance to say, look, for me to be part of this, this is what I think has to be done to this. And then we can talk through that, but it's nothing personal. This is just, these are what my constraints would be, just so you know in advance that, you know, that tension point will happen. And it's not, again, it's nothing personal. That's just scientifically, I'm going to have to think carefully about what I would want to put my name on in this context.
Absolutely.
Well, I think that but if at the same time, if this can't be written in a way that passes your sniff test, then we failed, right?
I think that's true, but whether I'm good enough to be the right sniff test is another issue that I think people could dispute at that point. But I do think that's true. I do think that I have the right opinion.
Yeah. So this has been just a tremendous amount of fun, and I really appreciate everybody participating. It's been a great discussion, and it sounds like we'll be able to get a transcript of it, since it's been recorded. And thank you all very much. I hope you all have a great day.
Yes, thank you so much for joining us. I want to remind everybody today, if you would like to continue this conversation, Jim Shapiro's Q&A is at 2 p.m. Eastern today. So you'll obviously get the normal 1 hour reminder for that. And yes, absolutely, this will be transcribed. It will also be added to the YouTube channel, which includes all sessions and all Q&A sessions. It may take a moment to get it both transcribed and edited, but we will absolutely do everything in our power to make sure that that happens. So I hope to see all of you later today at Jim Shapiro's Q&A session, and we really appreciate all of the wonderful synthesis of ideas that's happening here. I've got to say, even from a non-scientist point of view, it's absolutely fascinating. So I appreciate you all.